Hey everybody, thanks for joining me. I'm here today to show you how to dissect a sheep brain. There are a couple of different ways to do that. I'll show you two of them, though there are others. And I'm going to point out the structures so that you will be able to better understand what you are going to see when you dissect your own. So put on your thinking caps, and if you follow these tips, I know you'll be a cut above the rest. For this dissection, these are the materials that you'll use. Mosquito forceps for putting on the scalpel blade, some small scissors, a dull probe, a scalpel handle. Today I'm using a number 4 handle, which means I'm going to either need a number 20 or number 22 blade to fit this handle. And some forceps, our dissection specimen, and the dissection pan. Now I'm going to begin by putting in some paper towel into the bottom of the dissection pan and removing the specimen from its bag. Now, some terms about orientation. This would be a dorsal view. This is a ventral view. This would be anterior. This would be posterior. In the middle, if we were to cut down the middle, which we will later, that is called the sagittal plane or a sagittal cut. If we were to cut across here, like we are going to later, that's going to be a frontal cut. If we were to cut across here, that would be a horizontal cut, or a cut along the horizontal plane. Now, we have ordered these with the protective covering that goes around the brain and spinal cord called the meninges. When someone gets an infection of their meninges, that is called meningitis. There are 3 layers: the dura mater, the arachnoid matter, and the pia mater, which is closest to the brain. The dura mater is the one that is the thickest and is meant for protection for the brain and spinal cord. We're going to remove that. The other 2 are so tightly connected to the brain and the blood vessels going into the brain that they typically stay on and are difficult to completely remove without ruining the brain. So we are just going to remove the outer dura mater to be able to see everything. The other way that you can choose to dissect this is to leave the dura mater on and do your sagittal cut first, and then remove the dura mater. I'll show you that on a second specimen after this. The best way to remove the dura mater is going to be with the dull probe, the scissors, and the forceps. Now be careful as you're doing this to preserve all of these structures on the bottom. So here, the olfactory bulbs, I needed to make sure that I didn't accidentally cut those off. And for right now, I'm only going to be removing the meninges to this point and keeping a lot of these structures on the bottom of the brain intact. Now the last part to remove is going to be this slightly harder bony part that is in the transverse fissure that separates the cerebrum from the cerebellum. And to do that, you're going to very carefully move them away from each other and use your scissors to snip in a little bit at a time on either side, move them a little bit more until you can remove the entire thing. And there you go. There's still some dura mater here, covering a little bit of the brainstem. And the reason I kept it on is because this way you can see this little thing. This is the pituitary gland. Other things you can see at the bottom here, the optic nerve. So there would be nerves here that would go to the eyes. And the nerves cross because they go ultimately to the occipital lobe, but to the opposite side of the eye that it's connected to. So this would be the animal's left eye, but it goes to the right side of the occipital lobe. And so where they cross is called the optic chiasm. Chi is the Greek letter that looks like an X, and so it crosses here, the optic chiasm. Other things we can see here, we can see the spinal cord. The medulla oblongata is this first bump. This second bump being the pons. The midbrain is underneath here, and that's why we're going to remove this in a moment to be able to see that. We're going to remove the pituitary gland and these accessory structures. The olfactory bulbs are here that are responsible for sense of smell. You can see that they separate a little bit. You can see the frontal lobe here. The temporal lobes here and here. Again, the temporal lobes here. The frontal lobes here. The parietal lobe is this general area from here to here, and the occipital lobe being the very back. We can also see the cerebellum here, the longitudinal fissure, the transverse fissure. You can note that the cerebrum has many ridges and grooves. The grooves are called sulci, which is plural, or sulcus for a singular one. The ridges, the parts that stick out, are called gyri, or gyrus, for singular. What I'm going to do next is I'm going to carefully remove the remainder of the dura mater, which with it is sadly going to remove the pituitary gland, but it will let us see some structures beneath. To do that, there are a lot of blood vessels and nerves that are going through the dura mater that are going from the brain out of the dura mater, and so we're going to have to cut those to remove this. And so you gently pull away the dura mater and see what is going through it and make the cut. And so now you can see the complete brainstem, the medulla oblongata, the pons, and the midbrain. You can see the rest of the optic chiasm, how this optic nerve would cross and go here into the brain, and this one would cross here and go into the brain. This structure, which was where the pituitary gland was connected, is the base of the hypothalamus. Off of the side of the pons were large nerves that I had to cut. Those were the trigeminal roots. The brain has openings in it, as does the spinal cord in the middle. And the openings in the brain are called ventricles, just like some of the openings in the heart, and they contain cerebrospinal fluid. Here, when we lift up the cerebellum from the brainstem, in here is an opening called the fourth ventricle that is full of cerebrospinal fluid when the animal is alive. Now I'm going to put on my scalpel blade at this point. And the purpose for that is I want to cut a little bit off of the spinal cord to show you what a cross-section looks like of freshly cut spinal cord. If you look closely, I'm not sure how the color is coming out here, but you can see that the inside and the outside have different colors, that there are grooves that go in on both sides. This would be considered posterior, this would be considered anterior for a person. And there are different horns, and so there is the posterior horn where the inside structure goes to the posterior end, and anterior horns where it goes to the anterior end. Some more structures are clearly visible on the ventral side now that I've removed some of those accessories. This bulge here is separating the temporal lobe from other structures. The fissure here is called the renal fissure. These ridges are part of the hippocampus, which is the main structure that helps to consolidate short-term memories into long-term memories. So those are here and here. Structure up here is called the uncus, also involved in memory and emotion. And in the middle here, these are called mammillary bodies. When I remove the meninges, the arachnoid and pia matter stayed intact, and so I'm going to gently cut through those so that I can pull this apart a little bit to show you some things. Now, when you really pull the cerebellum away from the cerebrum, you'll be able to peek in, and you can see the superior and inferior colliculi. That's plural, and so singular would be the superior colliculus and the inferior colliculus. You also can see the beginning part of the fourth ventricle, just like the ending part of the fourth ventricle is here. So at this point, I'm going to do the main cut for this dissection, and that's going to be a longitudinal cut, which is going to go along the longitudinal fissure, but continue through the cerebellum, the brainstem, and go straight down. It's going to be important that you don't cut too deep with each single cut so that you don't tear the specimen with the sides of the scalpel handle. You want to be just using the blade to do the cutting. Now there we go. We've got two halves. One half might look a little bit better than the other, and so you can choose which one to get a photograph of. And we can see the inside structure of a lot of the things we saw on the outside, plus some new structures. The structures we already saw would be the frontal lobe, the olfactory bulbs, the parietal lobe, the occipital lobe, the cerebellum, the superior and inferior colliculi, or the superior colliculus and inferior colliculus, the spinal cord. It changes color once it turns into the medulla oblongata. The pons is a little bump here. And then the midbrain goes until here, where it changes color. We saw all of those on the outside, as well as the optic nerve. This was part of the optic chiasm that got cut. The things that we can see now that we could not see before, we can see the pineal body or pineal gland, which helps to control sleep-wake cycle. We can see this very inner ridge before the cuts started being made. This very inner ridge is the cingulate gyrus. Just below that, where it is white, is the corpus callosum. Connected to the corpus callosum and underneath it is the fornix. The area here is the thalamus. The area between the thalamus and the optic nerve is the hypothalamus. And then connected here was the pituitary gland, which got cut off before. On either side of the brain here, between the corpus callosum and the fornix, there's an opening. And again, an opening is a ventricle that would be filled with cerebrospinal fluid. And these are the first and second ventricles, one on each side. The third ventricle is here between the fornix and the thalamus. And then the fourth ventricle here, we've pointed out before, is here and comes out here. Notice the pattern of the gray and white matter in the cerebellum looks like a tree, and that's why it is called the arbor vitae, or tree of life. Most of the time you won't cut it perfectly in half, and so on one side or the other you will see a membrane that is connecting the corpus callosum to the fornix. You can see a little bit of it here, but for me, it mostly ended up on this side. And that thin membrane of tissue that separates one half of the brain from the other is the septum pellucidum. Here, this area that's open that would lead from the ventricles here to the ventricle here and carry the cerebrospinal fluid is the cerebral aqueduct. And now what I'm going to do is I'm going to make a frontal cut in one of these halves to show you what the cerebrum looks like, and you'll see the difference between gray and white matter. Now what you can notice about this is that gray matter is on the outside and white matter is on the inside. Gray matter is the cell bodies of the neurons, where the nuclei are. The white matter is white because of the protein and lipid combination myelin that insulates the axons, and so axons are all of the white matter. Now with the other half, I'm going to be carefully removing the cerebrum so that we can see what the surfaces of the interior structures look like. You can see where they separate. I'll be separating them both by pulling and using a scalpel. And as you do this, you'll notice that the corpus callosum really does lead into that white matter of the cerebrum. And so here we can see a lot of white matter, and those were essentially the axons, the wires that were coming from the cerebrum and going through the thalamus, which we can now see the edges of. The thalamus is a paired structure. There's one half on one side and one half on the other, and it is helping to relay messages between the brainstem, the cerebellum, and the cerebrum, which I just removed. The last thing that I'm going to show you is how you could have bisected this with keeping the meninges on and then remove the meninges after. Now the reason you might choose to do it this way is to keep the pituitary gland intact so you can see where it is connected to the brain. The pituitary gland is not technically a part of the brain, but is connected to it to the hypothalamus. And so now here we can see that the dura mater was covering everything, and you can see the pituitary gland here connected right to the hypothalamus. The pituitary gland and the connection to the hypothalamus right here. The thalamus, the hypothalamus, and the pituitary gland. If you chose to do it this way, you would then remove the meninges to do your exterior labels. When you are done your dissection, whichever way you chose to do it, removing the meninges first or removing them afterwards so that you could see the pituitary gland connected to the brain, you'll dispose of your things, put everything into a disposal bag, whether it's a large one for the class or a small one for individual groups, and rinse everything out. Now, wasn't that a lot of fun and educational? I hope that you had as good a time I did. Good luck on this, and I'll see you next time.